Good morning and good afternoon, everyone. So this is Tom Gao. I'm from the Scripps Research Institute, the YES lab. And so today I'm going to talk about the learning from deep learning, so a database search engine for feature identification. So let's start. So one of the central problem in proteomics is peptide identification. I think I don't need to emphasize too much to this group of audience. That's this is a very important problem. And so essentially the problem is how to get the sequence from the spectrum. And so you have one acquired MSMS and you want to get the sequence. How are you going to do that? So so let's review how the whole proteomics, the bottom-up proteomics works. So first you have a protein, and you use the first step is to use a protease to digest it into fragments. So digest it into tryptic peptide. If you use trypsin, that's the most used protease. And once digested, these tryptic peptides will be separated and then sampled by the mass spectrometer, and then we get the MSMS spectra of peptides. And the mostly used search algorithm for this is the database search engine. So the database search engine works like, so first we have a protein database. A protein database is all the protein sequences and that's known before. So we can get this piece of data from either sequencing data or the previous mass spec data. And then we can perform an in silico tryptic digestion to have all the tryptic peptide sequences. And then using the in silico fragmentation algorithm, we can now generate the theoretical peptides patterns, so a theoretical spectrum. And then we're going to try to match the theoretical spectrum to the actual spectrum. So and when we find a match, then we know the sequence. So this is like how it works in bottom-up proteomics. So there are three different types of peptide identification algorithm used for this problem. So the first of all, the most natural one is, I think, is the spectral library search, which essentially is you have a peptide standard and you acquire a spectrum of that standard and you compare with the one you obtained from the unknown sample. And if there's a match, so if the two spectrum matches, then you know you have a very good confidence that the one you just saw is actually the same as the standard. The problem with this is that obviously you need a standard for each peptide, and because there's many tryptic peptide, the possibility are very large. So this is almost impossible to cover like all the possible tryptic peptide in any species. So although there are efforts towards this end, like the human peptide atlas program, so try to do all the synthetic, try to obtain the information on like a consensus of the most abundant like peptides and to get a good spectral library for this. There are still problems, and for other species, like species other than human, there's not enough data. And also for each different instrument type, you need a new spectral library. For example, so if you have a spectral library obtained from ion trap instrument, you cannot use that spectral library directly to match the TOF instrument spectrum. And that will be a mismatch because different instrument will give you different type of fragmentation pattern. So, but the advantage of spectral library search is that it is more sensitive and also more specific because it's actually the peptide standard, and it gives you the exact fragmentation as you will get from the actual sample. So the most used algorithm for peptide identification nowadays is the database search. So the database search takes the sequence from a database, which can be obtained now by RNA-seq, And the advantage of that is obvious. So nowadays, for any given organism or species, you can easily obtain the sequence, like the RNA-seq or maybe even RiboSeq data for several $1,000s and within a week. So once you have this sequence data available, now you have a database, and then that's pretty much all you need. So with this database, you can do an in silico tryptic digestion or any other type of digestion. Say you use the chymotrypsin or you use the lyc, so you can do an in silico digestion using the pattern of chymotrypsin instead of trypsin, and so you can change your enzyme easily. And then from the tryptic peptide, you can perform in silico prediction. And once you have the predicted spectrum, you can try to match that with the acquired spectrum. The last one is the de novo search, which is shown here. So de novo search is actually very different from the previous two. So de novo search is a rule-based kind of system. So you have, once you have the acquired MSMS spectrum, there's no need to have any kind of prior knowledge of the database or the sequence, and it performs like it looks at the pattern of the spectrum and the mass differences, and based on the internal rules, it try to predict this without any database. So that's the de novo search. The advantage of de novo search is that obviously you don't need any sequencing data, although it's not very hard to get nowadays, but still some of them are not so easy to get, like the microbiome and some of the other, like a metaproteomics. So however, the de novo search is still in, I think, in an early stage, so the accuracy nowadays are not still not compatible with the database search. So ideally, so the perfect search algorithm or the peptide identification algorithm should be very sensitive and specific. So it has the sensitivity and specificity of a spectral library search, and it has the accuracy and coverage of a database search. So it can cover lots of spectrum without having a huge spectral library. By the way, a huge spectral library is very expensive because you need to synthesize all these peptides and sample them by different type of mass spec. And also it should be fast and use no prior knowledge. So it's best if you use no database, so you are not this database dependent. So toward this end, I think the key is intensity prediction. So if we roll back one slide, and what I mean by that is that in an ideal situation, if we can predict the intensity perfectly of all the B ion and the Y ion, essentially from the database, we can now have a spectral library. So if we can predict so a perfect prediction of the intensity basically will give us almost the same spectral as the one that we get from a non-peptide standard. So then we have all the advantage that the spectral library has, which is the highly sensitive and highly specific identification. So here I show you the on the bottom of the slides, I'm showing that so here is the actual this is a low res mass spec spectrum acquired in 1994 for this peptide, and this was published together in the Sequest paper. And in the middle is the prediction, like the in silico prediction made by Sequest. That was like a 1994. These two are published together. On the right is the prediction made by this program called MS2PIP, published in 2013, which is almost 20 years later. So I'm just showing that, you know, after 20 years, we can actually do a pretty good job in predicting the fragmentation pattern, like including the intensity. I mean, this is not a very fair comparison. I just want to show that we can now do such things. And this paper, the MS2P, actually, I think is the first one showing that we can use a large identified data, existing data, and train a model to actually predict intensity. So, and so with this in mind, here's what we did. So what we did is basically we trained a convolutional neural network for the prediction of intensity. So for any given sequence, we want to predict a theoretical spectrum with the intensity and so the data set used is the historic data that we acquired in the ESLab. So we have we acquired like 5 different type of instrument and fragmentation and including HCD, CAD, ETD, and we have 2.2 terabytes of SQT file. That's an identification file altogether. Covers more than 300 million spectra. So let me explain the convolutional neural network first. So a convolutional neural network is designed for image processing. So there are many layers. Now they call it like a deep learning. So deep basically means they have multiple layers. And so every 2 layers, they perform a convolution, then a subsampling, and then a convolution and subsampling. So before it gets all fully connected. A convolution means in image processing, a convolution means you can adjust like a contrast, you can do a compression, you can do a average and stuff. And these are for image processing. So we think for this piece of data, like to predict the intensity from the sequence, the convolution should be something has a physical meaning. So we actually predicted like a pKa, C log P, a charge, the M over Z, Pi, and the mass differences together with all the intensity data within the MS2 to do a convolution. So what we did is we did a 5 to 7 amino acid sliding window. So we start from one end of the peptide, and then we take the first 5 to 6 amino acids, and then we calculated the pKa, C log P, all these physical properties, and we put it into the first first position of this the first layer of this CNN, and then we slide one peptide to the right, and we put another one. And after we construct the first layer, we perform a subsampling, and then we have the second layer, and then we repeat this this process. So I'm not going to talk too much detail on this. So this is actually not very hard to implement, since we use the TensorFlow that provided by Google. Is all open source, and it's actually easy to use. So we implemented this one, and we trained it. Then with the help of GPU, we trained the data with a reasonable time. So each training takes a few days. And so here's some of the examples. Okay, so here's the result. So in this slide, the red one is the predicted spectrum from the sequence, and the blue one, the actual acquired spectrum. So as you can see here, the red one, the predicted one from sequence, directly from sequence after we trained the network, actually are doing a quite good job to actually representing the to reconstructing all the intensity information, even for the small details, as shown in the middle here. You can see that the accuracy is actually pretty good. So this is just one example, and we perform analysis of like how accurate this is. So when there is a peak in the acquired spectrum, and when this peak is within 15% of the predicted intensity, and if 85, we call it a successful identification of this peak. And when 85% of the peak find in the acquired spectrum is found to be correct, we call this whole thing a successful prediction. So here I shows the prediction accuracy curve for this trained CNN network. Because we started with at least 5 amino acids, so our predictions start from 6 amino acids. So the sequence needs to be at least 6 amino acids. And as you can see here, because we use a sliding window of like a 5, 6, or 7 amino acids, so the minimum. So in order to get like 80% successful prediction, we need at least like 10 amino acid, and we get the best result after 12 or 13 amino acid. So after the length of 12 or 13 amino acid, we can do a 90% successful prediction on longer peptide. For shorter peptide, if the peptide consists of only 6 amino acid, we can only do a 50%, around 50% successful prediction. When it's 10 amino acid long, we can do almost 80% for charge 2 and 3, and 65% for charge 4. And as you can see, the charge 2 and 3, they overlap. So the peptide sequence with charge 2 and 3, they overlap pretty well. But for peptide sequence with charge 4, there's a significant drop in prediction accuracy. So, well, we think this is due to the fact that the charge 4 spectrum are actually only like less than 1% of our trained data set. So our trained data set consists most of charge 2 and 3. And so we think the charge 2 and 3 get enough training data, and the charge 4 didn't. I think the charge 4 prediction can be improved upon training of more data. So with this trained convolutional neural network in hand, because we didn't use meaningless convolution like the contrast or some random convolution, we used actually the physical property prediction or calculation. We want to learn like what we can actually learn from the weighted node of this CNN network.
Can I ask you a question?
Sure.
What do you think in the previous slide? This is Salvatore. Why do you think your prediction improves substantially with the size of with the length of the amino acids?
With the length
of the peptide.
With the length of the peptide. So you mean what's the reason?
Yeah. Why do you think that is the case?
So, well, I mean, there are different. I think that there are different guesses, but my guess is that since we use a minimum sliding window of like 5 amino acids, and so if you look at the previous slides here, we use a sliding window from here, and then slide each slide one by one amino acid, and the sliding window is around like 5 amino acids. So it needs some in order to get a sequence of data like 5 data points, you'll probably need 10 amino acid, and in order to have like 10 data points, you need almost like 15. So that's one thing. Another thing is that So in our training data, there's actually a bias. So the bias is not introduced by us, is just the naturally tryptic digested peptide, the length, which can be sampled by the mass spectrometer. There is so the average or the most frequent seen length of the peptide is around like 12 to 13 peptides in the in our training data. So I think that could be the other reason. But for now, I don't know which one contribute more on this accuracy.
Right, thanks.
Okay. So so when we look at the nodes, so after training, we now have a fully trained CNN network, and we can look at each node, what's the weight of it and what this node mean. We can trace back to this node, and we find that what very interestingly is actually we can learn from this deep learning neural network. So for example, we find that the B1 ion is actually very rare because the nodes representing the B1 ion's weight on the intensity is almost zero, and that's indeed true. So for most of the CID and HCD fragmentation, B1 ion, because the fragmentation mechanism, B1 ion is actually pretty rare, and the B2 is much more dominant in the MSMS. And also some other things we learned is that when there's a proline, then the Y ion from the head means the Y ion from the left side of this proline is very dominant, and we that's also consistent with our previous experience. Excuse me. And there are some other things we can learn. For example, when there's a proline, then the B ions, there's a higher percentage of B ions. There are almost like endless rules that we can learn from this network. However, many of the rules doesn't actually make sense to, still doesn't make lots of sense. But we're trying to understand it, like what does it mean. But I think what we show here is that we can actually learn from this trained neural network. And I think the key here is to use a meaningful convolution, which is not just use contracts or mirroring or simple averaging, but to use some physical property calculation, which actually have a physical meaning in here. So with this piece of trained neural network in hand, now we can predict the intensity from sequence, and we want to see if this actually improves our identification. So this was developed together with a new algorithm that use a DFA or deterministic finite automaton. So the DFA is actually that how in computer science, like how the regular expression is actually searched with a string. So for example, if we want to search a string that's like A, B, 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 A, we can search like A and B, B and plus A. That basically means we can have a match of all the A, B, B, A, B, 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 A, and A, B, 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 A. So all these things that matches A followed by 2 B, B, and so on, and end with an A. We can match all these things, and that will not match. So A, B, A. So with A followed by 1B, you only match like A with a double B. And the above regular expression, the A, B, B, A, can be drawn as a DFA graph, as I showed like on the left corner here. So the important thing, like the reason why we want to use this DFA algorithm is that this algorithm for searching for matching the string. This is a linear algorithm which can match things that without knowing how many repeated patterns are in between the some of the known pattern. So, for example, if we have a sequence that we know the sequence consists of a DLE and then CGVR, but we don't know if there's a PTM on the N terminus or C terminus, and if we don't know like if there's a PTM on the E here, I mean a true blind PTM search, then we can use DFA like this, and this will give us our answer from a given spectrum in a linear time instead of exponential time. So essentially this give us advantage on speed to search a complex spectrum like the spectrum with PTMs or the spectrum in DIA data, which is the multiplex spectrum. So here's one example of the Dfa search. So when we have a synthetic complex spectra, what I mean by synthetic complex spectra is that it's not actual DIA data, it's actually the DDA data and already identified. So we combine 3 known DDA data into one, so representing one piece of spectrum which has 3 peptide fragmenting at the same time. And then we try these algorithms to see if how it performs, like versus the traditional database search engine. And this Dfa search can actually predict, can do a very good job deconvoluting this complex mixture of spectra. So as I shown here, there's a ROC curve for peptide IDs from complex spectrum. That's a synthetic complex spectrum. So and the y-axis is the fraction of correct identification, and the axis is the FDR. So first, let's look at the traditional database search engine, which what we use is the Perlucid, developed by the YesLab, and Perlucid treat this. Perlucid cannot search for more than 1 peptide per spectrum, so we treat it as like just 1 peptide per spectrum. And here's the FDR curve. And what we showed here is that our Dfa search algorithm can do this a similar job as Perlucid with the similar ROC curve. And however, when there's a 2 peptide or 3 peptides per spectrum, then the Perlucid doesn't work, and our Dfa search algorithm still works with a lower ROC curve. I mean, this obviously needs some improvement, but at least for now, this thing works, can decomplex a complex spectrum. So when the Dfa search and the intensity prediction altogether, when we put them all together, we can find that these two greatly accelerate the blind PTM search from a complex spectrum. So here's a comparison of the total search time by number of PTMs. So on the y-axis is the total search time, and you can see the traditional database search, when you increase the number of PTM within the search time grows exponentially. And if you compare it with our new search algorithm with the DFA and the intensity prediction, in the green curve, so the CPU versus CPU to Perlucid, it increases linearly to the number of PTMs. So also we can also accelerate this by GPU, and it performs the same, just a little faster. So here's the comparison, like of the total IDs with like 20 PTMs. So in Perlucid, we identified in total like almost like 120,000 peptides. Sorry, not peptide. That is a PSM. 120,000 PSM, and with the Dfa search, we identified almost the same and a slightly more than Perlucid. with the same data set, and when we look at only the PTMs, so the peptide spectrum match with PTM, the DFA search did a much better job, identified almost 15,000 PSMs with PTM, while Perusid were only able to identify around 9,000 PSM with PTMs. And here's the total number of PSM comparison with 20 PTMs, and you can see the new search algorithm with one peptide per spectrum actually identifies more than Perusid. And as we showed here on the upper right, most of these additional identifications are due to PTM that missed by the old database search algorithm. Okay, so in summary, so I think the computational power provided by modern hardware now allows efficient training of large and deep convolutional neural network, and that gives us a chance to train massive data through the CNN network, and to accurately predict B and Y ion series intensity from a peptide sequence. So the accurate intensity prediction improves sensitivity and specificity of the peptide identification, although there are more work to do to actually show that this is more sensitive and specific. And the accurate intensity prediction also accelerates peptide identification. There's still a large room for improvement, like training with data from different type of instrument, different species, and obviously for some of the type of the instrument, we need more training data. So eventually the goal is to have a database independent neural network and can predict the sequence as well as spectral library. Although I don't think that's entirely possible, but we'll keep improving. So I want to thank Professor Zhang Yates for providing all the support, and Bruce Cox for his support on the DFA and advice on the DFA implementation, and all the YES Lab members and the funding agency, the BD2K initiative, and NIH.